I think about her. Good morning, everyone. Okay. So, dear partners, dear colleagues, dear students, and all the attendants, we are glad the Institut Pasteur de Tunis for the 4th Bioinformatics workshop titled Leveraging Functional Genomics and Bioinformatics to Enhance Genome Editing for Disease Therapy and Drug Design in the frame of our GARN CLS project, which stands for German Arab Research Network for Computational Life Science project. I'm also glad to invite and to welcome our General Director at the Institut Pasteur de Tunis, Professor Semi Amnif, to give some welcoming words and open this event. So please welcome our General Director, Professor Amnif.
Thank you, Kais. Good morning, esteemed partners from Germany, Morocco, Lebanon, and Egypt, honored guests, distinguished colleagues, ladies and gentlemen. Good morning and welcome to Pasteur Institute of Tunis. It's both an honor and a privilege to open this international workshop dedicated to strengthening international academic and scientific cooperation in the interdisciplinary field of computational life sciences. The field of computational life sciences offers transformative potential for understanding biological systems, designing innovative solutions, and addressing global health and environmental challenges. Our workshop brings together leading research, practitioners, and experts from across the globe. This diverse assembly of talent and knowledge is a testament to the power of collaboration in driving scientific progress. Through this network, we aim to build bridges that facilitate not only academic and scientific exchange, but also enduring partnerships and inspire innovation. Pasteur Institute of Tunis has long been a beacon of excellence in scientific research and public health. This workshop represents a significant milestone in fostering international collaboration and advancing scientific innovation in the service of human health. I would express my deepest gratitude to all who have contributed to making this workshop possible. To Dr. Ghedira and the organizing team at the Institut Pasteur of Tunis, thank you for your dedication and hard work. To our esteemed participants, thank you for bringing your expertise and enthusiasm to this gathering. Your presence enriches our discussions and inspires our collective efforts. I wish you all a productive, insightful, and inspiring workshop. Thank you.
Thank you very much, Professor Semi Amnif, for these words and for the welcoming. I think that we can now start our program, and we will start our first session of very insightful presentation, and I will welcome our moderators. So Professor Dr. Rahma bin Abdul Razak and Professor Dr. Usama Khamisi to moderate this session.
Good morning. It's a great pleasure for me to welcome you all to this international workshop. Just I want to give you an outline of how this session will be proceed. We'll have 4 presentations, and I kindly ask our speakers to respect the time, just to stick with our schedules for the whole day. And after each presentation, we'll open the floor for discussions. And for those who are attending online, you can type your questions in the chat. So for the first presentation, I invite Professor Mohammed Hamid from Germany University in Cairo and Rostock University, Germany. So Professor Mohammed Hamid is Professor of Bioinformatics and Computational Biology at the German University in Cairo and head of the Integrative Omics Analysis Group, Rostock University Medical. Mohammed does research in systems biology, bioinformatics, and biostatistics. Professor Hamid, the floor is yours.
Good morning, everyone. Hello. It's my pleasure to be here for the second time in Tunis, lovely country, very, very beloved persons. I'm quite happy to be again here, especially at the IPT, Institut Pasteur de Tunis, within the framework of the German Arab Research Network for Computational Life Science. And before I get started to give you an introduction or overview about the project, the GRN CLS, I would like to thank everyone who was behind this amazing workshop. You have no idea how those people spent efforts, time in order to come up with this workshop in a very, very short notice. So special thanks to Qais, Osama, and Reem, and all the organizers, and also the team of Qais. I'm calling this like Qais' gang, yeah? So Yasmin, Slim, and also Shema and Faryal. Probably Qais also has hired a new person, I don't remember. But really, I would like to thank everyone who is really behind this workshop. So again, we were here last year. I think exactly at the same dates for the third bioinformatics workshop within the framework of the German Arab Research Network for Computational Life Science. Guys, sorry, I think it needs to be charged. It says battery.
Okay.
All right, so what is GRN? The German Arab Research Network for Computational Life Science. So it's a 3-year initiative between Arab institutions, or Arab academic and research institutions, and also German institutions. So it gathers actually, the academic partners from Germany, namely Rostock University, and this is the main coordinator of the whole initiative. And this is coordinated and represented by me. And also Göttingen University, and this is represented by Dr. Anna Hauschild. And also Saarland University in the southwest of Germany, and this is represented by Professor Dr. From the Arab and MENA region, we have the American University in Beirut, represented by Professor Hebal Haj. And we have also 3 institutions from Egypt. The German University in Cairo, represented by Professor Salim, who's also here. And the German International University, represented by Professor Hamdi, she's also here. And from also non-university partner, we have the EGCOM Bio Community, which is a community or a scientific community for bioinformatics and computational biology. And this is head by Dr. Asma Reda, but unfortunately she couldn't make it to be here with us today. From Tunisia, we have IPT, and this is also represented by Professor Dr. Ghedira. And from Morocco, we have also IPM, Institut Pasteur de Tunis, and this is represented by Dr. Fawzia. So the idea behind this initiative is to establish a long-term collaborations between all these different institutions in Germany and in Arab lands for a long time in order to have a sustainable research cluster in the field of bioinformatics and computational biology, and not only actually in bioinformatics and computational biology, but it could extend beyond that. So the general term, this is why we named it computational life science. All right, so as I said, this initiative is represented or funded basically by the DAD, the German Exchange Association. And today, inshallah, I'm going to give you an idea about these 3 points. So the DAD TAZIZ program, which the GRN is funded under its umbrella. And also, I'm going to give you an idea about the activities of the GRN itself. And at the end, I will also give you, open the space for questions. And if you have ideas or thoughts, I will be glad to hear them. So what is the DAD TAZIZ program? So the DAD TAZIZ program or partnership was an initiative that firstly developed in Cairo in 2022. So the DAD wants to think about ideas to strengthen and to foster the collaboration between Arab institutions and German institutions, and also between Arab institutions and each other. This is why it comes from TAZIZ, and you know, TAZIZ in Arabic means strengthening, to نقوش or to extend, to make it more stronger. So from here comes the problem or the program, which is TAZIZ partnership. So TAZIZ program with DAD comes actually with 3 programs or 3 lines. The first one, it's called networks. And the second one is science cooperation. And the third one is short-term measures. So the first one, which is the networks here. So those are the main 3 pillars. The first one focuses on establishing or involving, you know, political parties and allies into the development of countries and so on. The second one, which is this red one here, focuses on fostering the scientific collaborations between the academic and research institutions. And the last one here, which is like a short-term measure, which is kind of a seed to establish long-term collaborations between the different institutions. So we started actually with the science cooperation. I'm going to tell you our history in applying for these funds. So we started with this one, and in 2019 till 2021, we have got a fund for empowering the bioinformatics research and computational biology in Egypt, and this is called EGCOM Bio. And an outcome from this project was actually the idea of having a community for bioinformaticians and computational biologists in Egypt, and expanding it later on to cover the whole MENA region. Besides, we have developed a complete program or complete diploma program for bioinformatics and research and computational biology research, and many of our alumni now are working and also studying or continuing their studies in esteemed institutions worldwide, including Germany, Europe, and also United States of America. Then the next one was the GRN-CLS, in which we are right now. So the German Arab Research Network for Computational Life Science, and as I explained, it connects different institutions from Germany, from Egypt, from Lebanon, from Morocco, from Tunisia, in order to foster the collaboration and also the interactions between these different institutions. Then, for the short-term measures, which is like small research projects, usually stays for like 3 months to 9 months. We started with the GED permit AI, so the German-Egyptian dialogue on tackling precision medicine using artificial intelligence, and that was also in 2020 between Rostock University, a German institution, and Nile University, that's a university in Egypt. So the second one, which was recently also acquired, it's the GenCap project. And here we have the main coordinator, the main German coordinator of the GenCap project, Dr. Amira Mitwali. And it stands for the German-Egyptian Microbiome Capacity Building Project. So trying to involve now the microbiome research and in all the applications of healthcare as well as also the industrial application. And finally, we have acquired also recently the German-Jordanian Research Cluster on Intelligent Healthcare Informatics. So this is also a collaboration project between Germany and the German-Jordanian University in Amman, in Jordan, in order to help them to foster the interdisciplinary research, which is intelligent healthcare. So the applications of artificial intelligence, especially machine learning and deep learning in healthcare informatics. All right, so as I said, the GRN-CLS is mainly under the umbrella of the second pillar, the science cooperation, which aims to establishing collaboration and expanding the collaboration, especially in research in the field of bioinformatics and computational biology. So the project actually was ranked #1 among all funded research projects in the database, as you see here. So they have funded 17 projects, and the GRN was ranked #1 in order to, as a mark for its importance and also collecting and gathering many partners from all different Arab regions. So you might ask, what will be the activities of the GRN-CLS? So we are in the second year of the GRN-CLS now. So let me give you a brief overview about what has been planned and what has been achieved so far. So we planned actually to develop a master curriculums in 2 interdisciplinary fields, bioinformatics and also molecular medicine. So we have got so far, the first point is done, and we have already made now international curriculum in bioinformatics and computational biology, and this is going to be applied first at the German International University as well as also the American University in Beirut. The second part, or the second activity, is actually to have short-term research visits and exchange as a kind of lab rotations between the different partners. And we have already applied this activity this year. So we have got Ferial, also one of Kais's team. So she's gonna visit our lab in the German International University in Egypt. And also we have got another student from the AUP in Lebanon. She has visited already our lab, specifically by Dr. Muhammad Salim at the German University in Cairo. In the next year, we plan also to have a student from Morocco, from IBM, but we didn't discuss yet the destination, so we are looking forward. Also, we have planned a lot of training events, as you see right now. So we have planned 3 international workshops. Today's workshop is one of them, as well as other activities like international hackathons, training hackathons, scientific hackathons, as well as also lecture series by top speakers in the field of bioinformatics and related areas. At the same time, we planned also to establish a panel of experts in multidisciplinary field to help all the partner institutions to apply or to help them in judging the promotions of their staff. For example, if you have a staff in the level of assistant professors, and then he or she needs to get promoted to the next level, like associate professors or full professor. So we have got this committee already established. So we have Professor Volkert Helmers in it, and also I'm also a member of this committee, and we can, judge on, the availability or the applicability of promoting any person related to, wants to get promoted to the next level, specifically in the area of bioinformatics and computational biology. So I would like to give you a brief idea also about what has been done and some clips of, our activities. So in 2023, we started with the first bioinformatics workshop that held, that was held actually in Lebanon. So as you see here, some clips and some, photos or images from our activities there. So we planned this workshop to introduce them and to put them on track in order to be able to talk the same language, because our partner is pure biologist. So we planned this workshop at the American University in Beirut. The second one was actually the main one, which witnessed actually the inauguration and launching of this network. So that took place at the German University in Cairo with the participation of all partners for the same time. This is the only event that witnessed the participation of every partner in this network. All next events, actually, we lost or we missed one or two of the partners. So that was a very, very big event, happened and occurred in Cairo at the German University there. Then, last year, exactly at the same place here, at the same hall, so we have organized the third bioinformatics workshop, which was under the title The Power of Data, thanks to Kais and Osama and all teams also. And we had really, really very positive feedback here. We discussed a lot of future events, as well as also we explored the avenues for collaboration between our partners and also other external interested partners. Then in 2024, this year, so we had our first activity when we organized the Biohack Hackathon. And this is kind of activities that's underrepresented in our Arab culture here. So to have, kind of teams, and every team is planned or putting an effort to solve and tackle a specific technical problem in bioinformatics. And we organized this event at the German International University in Cairo. And here you can see a lot of shots and images about this great event. And we have got more than 12 challenges, real 12 challenges. Every challenge was offered by a mentor, and this mentor was helping the students in order to help them or to tackle this problem and to present the solutions later on. It was a magnificent event and was one of the greatest events actually that we had within the GARN CLS network. And then we have got also the next event in this year. It was organized also by Dr. Fouzaira Rouhani and her team at the IPM, Institut Pasteur de Tunis. It was also one of the great events and very, very interesting experience we have got there in Morocco. And finally, the last event for today, for this year, is actually this event where we are right now. So the fourth bioinformatics workshop at the IPT, and we are sure it's going to be also a very successful event like its predecessors. Before ending my presentation here, I would like to focus on one important thing: the importance of having communities, scientific communities for a specific field. So we started by having EGCombio as a scientific community for bioinformatics and computational biology in Egypt, and it would be great to have this experience also replicated here in Tunisia, in Morocco, in Lebanon, and we are doing all our best to have these societies, scientific societies. You have no idea about the importance of having these societies. It helps a lot for knowledge exchange. It helps a lot for being internationally visible for other international communities in Europe, in the States, and in everywhere. And also it helps for opening communication channels between all different scientific societies. This was basically my presentation about the GRN-CLS network, and as I promised you, so we are all ears. If you have an idea, if you have one thought that you think it could be beneficial also to be implemented within the framework of the GRN-CLS, please talk to us. Yeah, don't be shy. Just offer it to us. And yeah, we still have 1 year of activities, so the whole 2025. And afterwards, we will be quite happy to apply all your ideas if we all agree that it's going to be beneficial. At the end of the day, I would like to thank all funding agencies behind the GRN-CLS network, basically the Federal Foreign Office or the Ministry of Interiors in Germany, and also the DAD, the Deutsche Akademische Austauschdienst, and I would like to thank all of you for your attention. Thank you.
Thank you, Professor Hamid. The floor is now open for questions from the audience. Just 5 questions, please.
Everything's fine? Okay.
Okay, so let's move to the second presentation.
Now we will start with our colleague, Professor Dr. Senior Biologist. Senior Biologist with the head of the GMO and member of the European Consortium. This consortium is made up of experts in STEM cell biology. مش في الفلاش الفلاش هذا لاش تعمل هذا قام فينا
قام فينا ليه قام فينا ودي يعقوب
هذا
يحط من البرسنتاتور
هي لحظه برك
زدت حاجات على
باش نحطوا ستريم
سي كونيكتي بالخارج
ديجا
اكستيريور
في ستريمينغ احنا يغزروا لنا كوكو
اسمع فينا
كايند اوف
صديق مشهور استنى حتى تثبتوا معي ما تمشيش
انت جاوبي
استنى قاعدة هنا
انت للحظه هي
شنو القصة دي؟ سي باش نطلع
Good morning, everyone. Thank you. I would like to thank the audience to be here, as it is a very cold day, so you are really a fantastic audience. I would like to thank my colleagues who introduced me, Dr. Osama Khamisi Daha. I would like to thank also my colleague Kaysar Tila for this kind of invitation. So, okay. For those who don't know me, I am Professor Ghedira from the laboratory Bioinformatics Genomics and Genetics at Institut Pasteur de Tunis. So it's a big pleasure for me today to present our work and main achievements about functional genomics and cellular modeling for rare genetic diseases to unveil associated pathogenicity and novel strategic therapy for these rare diseases. So what is a rare disease? It is a pathology that affects less than 1 in 2,000 individuals in the general population. There is about there is about 7,000 rare diseases in the world, which affect 6 to 8% of the general population. About 80% of genetic origin. The majority, about 75%, affect children, and 30% of affected children die before the age of 5, unfortunately. So these rare genetic diseases are frequent in North Africa and in the MENA region as well. This is mainly due to the high rate of consanguinity and in Tunisia, for example, there is 589 genetic diseases which were reported by our lab, of which 60% are autosomal recessive diseases, which were associated to 313 genes. The diagnosis of a rare genetic disease is sometimes very complex. There are 3 main requirements for an accurate diagnosis. Engage a team of healthcare professionals to address various aspects of these diseases. Seek out for genetic specialists and disease experts, and also seek out for advanced diagnostic techniques as WES, whole exome sequencing, target gene sequencing, or whole genome sequencing. So for the majority of these rare diseases, there is no dedicated treatment. What we are doing currently is tailored therapies. Clinicians use approved drugs to treat symptoms individually, as there is no cure for the disease itself. Or supportive care, for example, they prescribe physical therapies or nutritional support to improve some symptoms. The major challenges that we are facing to manage rare diseases are to raise patients' awareness about the disease and the management of this disease, encourage patients to participate in clinical trials to gain access to experimental treatments, promote gene therapy. It is a promising but costly option for many rare diseases, and promote nutri-therapies to improve clinical symptoms of some symptoms of these diseases. Another challenge specific to countries with high rate of consanguinity, as for Tunisia, is the comorbidity. Indeed, several cases that we studied in the lab were families with comorbidities, as in this example, where 3 pathologies were associated at the same time. There is, for example, the xeroderma pigmentosum, the ichthyosis, and a melanoma cancer in the same pedigree, as you can see. So, actually, we are still trying to overcome challenges of identifying genetic etiology of these rare diseases. That's why we increasingly use NGS techniques to diagnose these pathologies, given the complexity of the rare phenotypes, but costs remain the limiting factor. In the meantime, a transition from genetics to genomics through bioinformatics, transcriptomics, epigenetic studies began several years ago to better understand the pathophysiology of these diseases and to connect the genotype to phenotype. Indeed, the term genomics involves all these disciplines that allow determining genetic variants and giving rise to genomic biomarkers. These genomic biomarkers could have either a prognostic, diagnostic, or therapeutic role. That's why their role should be confirmed through computational analysis and through cell-based assay. These biomarkers will provide valuable information on the pathophysiology of the disease and serve as tools for monitoring patients' response to treatment, which is the ultimate step for implementing a personalized medicine. So this is our research strategy in the lab, where we try to integrate genomics and cell-based assays to promote research into rare diseases and pave the way for novel therapies. Here I will talk about 3 DNA repair diseases. the Xeroderma pigmentosum, the cocaine syndrome, and ataxia telangiectasia. In addition, we use these rare diseases, these 3 models, to elucidate more common diseases. For example, we use the Xeroderma pigmentosum to study early onset of skin cancers. We use the cocaine syndrome to study premature aging or accelerated aging by studying immunosenescence, skin aging, muscle atrophies, mitochondrial defects. And we use the model of ataxia telangiectasia to study neuroinflammation and the impact of exosomes. Indeed, the shared phenotypes between rare and common diseases suggests common pathways, common biomarkers, and sometimes the same treatment. That's why we use rare genetic diseases to elucidate the common ones. Here are 2 DNA repair diseases, the Xeroderma pigmentosum and the cocaine syndrome. Both are characterized by defects in the DNA repair pathway, the NER pathway. The Xeroderma pigmentosum patients are characterized by acute photosensitivity that predispose to skin cancer. Some patients display also neurological disorders. Regarding the cocaine syndrome, it predisposes to premature aging with the severe neurodegenerative disorders. And we were particularly interested in studying both diseases because for several forms of Xeroderma pigmentosum and the cocaine syndrome, these diseases are undiagnosed in the country. Here I show you a complex genetic scenario. The same gene is linked to different clinical entities, and conversely, several genes could give the same form of the disease. That's why it is very complex to study the genetic etiology and connect the phenotype to genotype for these diseases. Okay, so this is our general approach, which is a multidisciplinary approach to fix genetic etiology for both syndrome, the XP and the cocaine syndrome, using NGS technology, and to identify molecular biomarkers involved in muscle atrophy using muscle stem cells, molecular biomarkers involved in melanoma cancer using XP tumor biopsies, and molecular biomarkers involved in skin aging using cocaine syndrome skin fibroblasts. The ultimate goal of this approach is to elucidate the mechanisms behind the disease, connect the genotype to phenotype, and help implementing novel therapies. For the Xeroderma pigmentosum, several forms were identified in the lab several years ago. 5 forms were already identified before our work in the team. The disease is more frequent in Tunisia than in other countries due to the high rate of consanguinity. As you can see in the pedigree, there is, which I show here, the rate which is very, very high regarding the consanguinity in the country. It's important also to note that genetic causes of this disease are not always easy to find because we can find different clinical phenotypes, but the patients share the same mutation. So that's why sometimes it took a lot of time to find the mutation, link it to the phenotype, and that's why we use increasingly NGS now. Here is another example of a particular phenotype of Xeroderma pigmentosum. This is a sibling who presented mild neurological impairment. Both patients were often exposed to sunlight but had no evidence of skin cancer, which made clinical diagnosis uncertain. It took a lot of time to identify the genetic etiology, and thanks to the support of our collaborators with whom we performed targeted gene sequencing, we identified for the first time the XPG computation group in Tunisia and in North Africa. And it was with these 2 cases that we start exploring structural modeling of the mutated proteins to confirm the pathogenicity of the novel variants. Indeed, we received a case who developed cancer at the age of 15, which is quite late for Xeroderma pigmentosum, and for whom no recurrent mutation had been identified. So using NGS, we identified 2 mutations in the same gene, And in collaboration with our colleague Hossein Afben, we determined the causative variant among both mutations, which caused a distortion of the protein. And for a second case, this one, it was also a particular phenotype of Xeroderma pigmentosum. The patient was aged 58 years old, and it took a lot of time to find the mutation, and we didn't know why it was very mild phenotype, and it was because the mutation induced very slight steric clashes that do not affect the domain of the protein. So that's why the phenotype was very, very mild. So in a recent work, we used the XP model to study the impact of DNA repair pathway impairment on the expression of a set of genes in order to identify biomarkers that could be involved in melanoma cancer development. We studied the expression of 4 candidate genes involved in inflammatory pathways in melanoma tumors from XP and from sporadic cases. And we use qPCR sign array technology. And our findings were compared to those of public RNA-seq databases as for GTEx and GPL. And we found that, as you can see here, there are a specific transcriptomic signature for XP melanoma tumors and for tumors from sporadic cases. In addition, the majority of inflammatory genes, about 34 genes, were underexpressed in XP tumors, which suggests an immunodeficient status in patients with XP. More interesting, we showed that the kallikrein genes, especially the KLK6, was the most expressed gene in melanoma tumors in XP, which was associated to a worse survival of patients. And that could be used as a prognostic marker. The second model of rare disease that we studied is the cocaine syndrome. The patients are characterized by progressive neurological disorder, a progeria with appearance, microcephaly, and severe psychomotor delay, as well as tooth decay, as you can see in this photo. In Tunisia, only 5 cases were reported prior to our study. These studies focused essentially on the clinical aspects of the disease as bone dysplasia, and also a work who showed that the patient don't develop photosensitivity. And this was very strange because normally the patient with cocaine syndrome should develop photosensitivity. That's why we start a project, a collaboration with several hospitals in Tunisia, and with 2 partners from Paris and Strasbourg, as well as a team from Institut Pasteur de Tunis, the laboratory of hematology, to study this syndrome because it was very complex and undiagnosed in the country. And we found all the mutations that were involved in this disease. And also we showed that there is a huge clinical heterogeneity that, and also we find that there is no photosensitivity in some patients, which evoke that this disease, it is linked to a DNA repair pathway, but it's not the only pathway that is responsible of the disease. So genetic investigation that we conducted allowed us to classify the patient in 3 groups, and these mutations were their pathogenicity were verified by UDS and RRS tests, which are cell-based assays, the gold standard of confirming variant pathogenicity in this disease. And we, thanks to our work, we implemented prenatal diagnosis with our collaborators in Charnicol hospitals. But since its discovery, this disease, and the authors linked it to DNA repair pathway, but many studies suggest that it's not only linked to the DNA repair pathway.
and that there is other defects behind the disease, especially because there is patients who develop photosensitivity and other not. So, as you can see here, there are patients which are affected by UVSS syndrome, which is a syndrome who have the same mutations in the CSA and CSP gene, but don't develop neurodegenerescence. This suggests the involvement of other mechanisms. Indeed, both proteins, CSA and CSP proteins, are also located in the mitochondria, where they play a role of autophagy. Now, cocaine syndrome patients their cells contains also a high rate of ROS with redox imbalance. That's why previous studies indicated that the impediment UV response in these diseases exists, and there is other mechanisms behind the syndrome. In addition, our partner in Institut Pasteur Paris, through the project, a PTR project that we have together, showed that in the patient cells, the POLG1 was strongly reduced in the CS cells, and that the reduction was strongly linked to the severity of the disease. In addition, our partner showed that using an antioxidant molecule can restore the function of the cells. So this molecule that they tested was later classified as an orphan drug disease. Having understood some of these causes of the CS disease, we wanted to find out whether this model shared the same alteration as those of elderly. This would enable us to prevent age-related diseases. For this, we established primary cell culture from skin biopsies obtained from cocaine syndrome patients and from healthy donors, aged donors. And we studied the expression of 84 candidate genes involved in oxidative stress. Our data were compared to those of RNA-seq public data. And key pathways were further analyzed in a drug bank database to identify drugs or molecules that could impact these altered pathways. We found that patients with cocaine syndrome mutation have a transcriptional profile that is more similar to that of elderly donors, and that the transcriptional profile of the group CSB, who have more severe phenotype, cluster separately from the other group, the cocaine syndrome with the A form. In addition, we showed that the main genes altered affected two main pathways. The first one is the arachidonic acid metabolism, and which its disturbance can induce inflammatory defects, and the NERV2 pathway, which is involved in maintaining mitochondrial function. We found that So we found that patients with CSI mutation have this transcriptional profile that is distinguished, and it depends on clinical features. So that's why for these two pathways, we could impact them by other drugs that target the mitochondrial defects. That's why we use the nicotinamide. It is a water-soluble form of vitamin B3, and it was shown that this supplementation with NAD plus can improve lifespan and hearing loss in cocaine syndrome experimental models. We tested the nicotinamide on cocaine syndrome patients' fibroblasts, and it was interesting to see that POLG1 expression was partly restored, as well as the expression of some genes which was dysregulated in patient cells. This suggests that supplementation with nicotinamide could hold promise to prevent age-related diseases. Currently, we are studying the effect of nicotinamide on muscle regeneration using muscle stem cells. Indeed, this is a protocol that I started several years ago, which consists on isolating muscle stem cells and modeling human muscle in dish. Primary results showed an improvement of myofibroblast fusion, and thus enhance muscle regeneration and prevent muscle atrophy. which would be a promising therapy for rare diseases. The third model of DNA repair disease that we used is the ataxia telangiectasia disease. It is also a neurodegenerative disorder. There are mainly two forms, the AT classic phenotype and the AT variant phenotype. We classified the patient that we received and we find all the mutation using NGS technology. And in this model, we tried to verify the pathogenicity of some variants using structural analysis and molecular docking to confirm the clinical diagnosis and go further in our investigation. So we find 5 novel mutations. The majority were classic phenotype, very severe phenotype. And for 2 patients, there was they harbor a mild phenotype with missense mutations. So we try to elucidate their impact on patients' phenotype. So we collaborate with Dr. Ghedira and Dr. Oussama Khamesi. And a structural analysis based on modeling and molecular docking was carried out. This is the pipeline that was used by Oussama to evaluate the interaction of ATM with its main ligand, the P53. And both mutation, the missense mutation detected in the 2 different patients, increased the binding affinity of the ligand P53 to the receptor of ATM and increased the stability of the complex ATM P53, which could explain the pathogenicity of these variants. Indeed, the first mutation results in the formation of a more stable dimeric ATM P53, and this may affect the cellular response to oxidative stress. The second mutation induces a stable monomeric ATM P53 complex that can alter the transition from the active to the inactive state in response to DNA damage. This confirming these variants effects in patients would pave the way for personalized medicine for this patient with these specific kind of mutations. To finish, in parallel with our AT projects, we have recently set up a collaboration with the University of Salamanca in Spain to study the involvement of exosomes in the pathogenesis of ataxia using in vivo mice models and ex vivo models, organotypic culture. And more recently, a bilateral project was funded by the Tunisian Ministry of Higher Education and by the TÜBİTAK. It will start in 2025 in collaboration with the Izmir Biomedicine and Genome Center in Turkey to characterize microRNA from neuronal exosome in autosomal ataxia to unveil mechanisms to therapeutic insights. And to finish, I would like to thank all my collaborators, the donors, the patient support group, and especially my hard workers team. And thank you all. Thank you for the assistance.
Thank you. Are there any questions from the audience? We have time for questions.
Thank you so much, Dr. Ghedira, for this nice presentation. Very confident presentation. So I have 2 questions. One about the evaluation of the So 2 questions. One about the information of rare disease. This is about the incidence of the disease, the phenotype the phenotype form, the genotype form, or about the treatment that is really. And the second question is about the base of the gene for this treatment.
الو الو activate your microphone you can present now. طيب سامعني أسامة، سامعني كده كويس ولا ايه؟ انا مش سامعاكم. طيب انا المفروض اعمل ايه؟ انا مشغله المايك بس مش عارفه يعني، هو دلوقتي كل حاجه شغاله عندي بس مش عارفه انتوا مش سامعين الصوت. طيب انا المفروض اعمل ايه؟ انا مشغله المايك بس. الو ايوه اسامه، انا بتكلم بس انا مش عارفه انتوا مش سامعيني يعني كلكم انا اعمل ايه المفروض؟ طيب ايوه it's activated it's activated. اه ايوه اوكي، انا كل حاجه as activated والمايك يعني شغال ف اوكي. سامعني؟ سامعني أسامة؟ ألو؟ ألو؟ أيوه. اهم. أيوه أسامة. maybe أنتم عندكم السماعات مثلًا مش شغالة؟ لأن أنا أصلًا المايك شغال عندي حتى في الentire screen. طيب دلوقتي سامعينّي؟ ألو سامعينّي كده؟ أسامة ممكن تconfirm معايا؟ مش سامعينّي برضه؟ إمم. أنا معلية الvoice. أنا I clicked on the mic. Do you see المايك عندي شغال. هو شغال دلوقتي. إمم. إمم. أحطه؟ It's okay. السلام عليكم. طب انت بس أسامة كونفيرم معايا لو انتوا سامعيني كويس. اوكي. السلام عليكم. good morning everyone. في الأول حابة أشكر أسامة ودكتور قيس دكتور محمد حامد على الانفيتيشن الجميلة ديت إن أنا أكون بارت من جارنر وأشارك معاكم التوك بتاعتي النهاردة. أنا في الأول اسمي أسماء محمد رضا. I'm a postdoc at Faculty of Science and Faculty of Computers and Artificial Intelligence. Also I'm the president of EG-ComBio. عنوان التوك بتاعي النهاردة هيكون Decoding the Cancer Network and the Role of Hub Genes in Cancer Progression. في الأول هنتكلم على aim of this work. ده حاجة من recent work بتاعنا اللي احنا إن شاء الله we are going to publish. والaim بتاع الwork بتاعنا إن احنا عايزين نشوف correlation ما بين الانفكشن ببكتيريا اسمها H. pylori والgastric cancer. بس قبل ما نبدأ التوك بتاعنا النهاردة هنشوف يا ترى ايه هي المين causes of the H. pylori infection. فلو حضرتك بتعمل حاجة من الحاجات ديت اللي هي مثلا you usually eat fast food ودايما بتشير الفود مع الأذرز وبتاكل مع بعض of the same dish, or even you kiss your partner, or even you kiss your pet. So these are the common risk factor with the H. pylori infection. طيب دلوقتي الغرض الأساسي من الدراسة بتاعتنا دلوقتي إن احنا نسأل السؤال ده: How does the infection of H. pylori may lead to gastric cancer? بصراحة كان في two scientists كبار في المجال اسمهم بيري مارشال وجون روبن، أخدوا نوبل برايز سنة 2005 على الديسكفري بتاعتهم إن ال H. pylori كان lead to gastritis والألسر، والعلاقة ما بين ال H. pylori والجاسترايتس والألسر، ولكن ما زال اللينك ما بين الcommon disease signature leading to الكانسر is still unknown لغاية دلوقتي. وإحنا في البيبر ديت بنحاول نفك أو نعمل deciphering لللينك ما بين ال disease progression leading to gastric cancer under the infection with H. pylori. طيب الورك فلو بتاعنا جوه البيبر ديت. Okay, so the workflow in this paper is like this. Once the one is infected by H. pylori, we monitor the disease progression in 4 main stages: H. pylori infection and H. pylori gastritis, as you see with my cursor, and also H. pylori associated atrophy, then leading to H. pylori associated gastric cancer. To solve this problem, we actually collected data from a microarray dataset which represent the H. pylori infection and RNA-seq that represent gastric cancer, and then after that we identified the common tags and then we decoded the cancer network. بعد كده كان التكنيكال ورك فلو كان زي التالي: احنا جمعنا we collected 3 data sets representing the microarray data sets from GU and also the gastric cancer from TCGA. والورك فلو مشي بتاعنا كالتالي: we removed the least 10% variance gene, then after that we make the normalization and scaling for both samples and genes. and the analysis بتاعنا كانت ماشية بالطريقة الفلانية. for microarray dataset we performed lemma for microarray dataset, and for RNA-seq we performed the DSEQ2. Then we identified the differentially expressed gene and we represented using the heat map and volcano plot, and also we clustered the sample using the 3D PCA plot, and then after that we monitored the intersection between all study group, and then we decoded the cancer network using these plots, as I will mention after that, inshallah. So the first question in our paper was like that: how does the differentially expressed gene identification map the progression of H. pylori infection to gastric cancer? And to answer this question, actually, we performed, as I mentioned earlier, the lemma for analysis of microarray dataset and DSEQ2 for the RNA-seq data. And as you can see here, this is actually the results of the H. pylori infection versus control. and this is the heat map for the up and down regulated gene, and this is also the volcano plot that can show the up and down regulated gene, and this is also the 3D BCA plot that show the clear separation of the clustered samples. We performed this for the H. pylori infection versus control, and also we performed this for the H. pylori associated gastritis. Additionally, we also performed this for the H. pylori associated atrophy versus control, and also for the gastric cancer. To more confirm that these DEGs actually come or our therapeutic markers only come from the H. pylori infection leading to gastritis, and it's completely different away apart from the autoimmune gastritis, we also performed this analysis to confirm that our therapeutic and diagnostic markers come only from the infection with the H. pylori. After that, we asked the second question in this paper, which are what are the common DEGs in the disease progression that can guide the therapeutic biomarker discovery? And to answer this question, actually, we make the intersection between all data set that found in the study, like H. pylori infection, H. pylori atrophy, and H. pylori cancer, gastric cancer, and also H. pylori gastritis. And we successfully identified 43 common therapeutic markers that found within all data sets after our analysis. We also identified the common upregulated DEGs between all study and the common also downregulated, but we focused on the 43 one that are common through all studies. After that, we decoded the network between the common DEGs that were identified in the previous step from the Venn diagram and the upset plot. And then after that, we asked our third question, how does the network analysis of Go enrichment analysis reveal the key biological pathways? And to answer this question, actually, we performed the Go enrichment analysis for biological process and molecular function and also cellular component, and we identified actually key pathways that are common between all of these stages, like the inflammation, which can lead to the gastric cancer, and these are after decoding the networks of the common genes that are enriched in these pathways. Additionally, or in addition to that, we also analyzed the KIG pathway, reactome, and molecular signature database, as you can see from this. And then after that, we asked our first question in this paper, which is what are the key hub genes that found within the identified network? And actually, to answer this question, we performed this kind of analysis using 5 different algorithms, which are DMNC, which is a density of maximum neighborhood component, and also EPC, which is the age percolated component, and also the degree centrality, MCC, maximal click centrality, and also closeness, which is closeness centrality. And after identifying the hub genes from the 5 different algorithms, we took the common genes between all of these common algorithms. And then after that, we asked our 5th question in this paper, which are what are the role of hub genes in the cancer progression? And as you can see also from the analysis, these key hub genes, or the identified hub genes, or the common identified hub genes from the 5 different algorithms have a role in gastric cancer progression. As you can see, this is one example of our common gene, which is CXCR3, that is expressed in the 4 different stages of the gastric cancer, in addition also to the other genes. And then after that, we confirmed the expression of these genes in gastric cancer versus normal, and we find 4 of them are highly significant expressed in gastric cancer, like CXCR3 highly expressed in gastric cancer versus normal, also CXCR5, CXCL1, and also CCL2. In addition to that, we performed the survival analysis for both the normal and the gastric cancer patient for these kind of gene. And then after that, we come to one of the most important figures here in our paper, which is the investigation of drug-gene interaction. After decoding the most important gene, or the 10 genes that are commonly found between all data sets representing the disease signature after infection with the H. pylori leading to gastric cancer, these are actually the common gene. And we tested the drug-gene interaction, and actually these are the most important drugs that can inhibit and bind effectively to these gene, and this is actually the binding score. And here is our selling point for this paper. When we explain this paper to the clinician or the physician, we mentioned that if you find these therapeutic markers are upregulated in the disease progression of H. pylori, you can give such kind of medication which are FDA approved to treat these kind of disease. And also for the medicinal chemistry or the drug design scientists, these are also some of our upregulated genes in, in the study, and these are actually the drugs that can interact with these genes, and also with the combination with the binding score, as you can see here. But these drugs are still in the developmental stage, and they still didn't reach to the approval stage, so they can work more on these drugs until they can reach the FDA approval. In addition to this, we wanted to actually see the expression of these digs through the disease progression. We don't want to collect the data from separate studies like infection, like gastritis, like atrophy, like inflammation, or even gastric cancer, and do the analysis one by one. So we decided also to do what is called combat integration, to integrate between the whole data set. So in one single expression matrix, we integrated between the 3 microarray data set and the RNA-seq data to see the trend of these genes through the disease progression. And as you can see here, this is actually the control, and this is the H. pylori infection, and this is also the H. pylori gastritis, and this is the atrophy, and this is the cancer. And as you can see here, this is the trend of our markers that go from the normal or the control, and then upregulated until reaching the cancer stage. So our results are confirmed whether on the scale of analysis of single expression matrix, or even combine them using combat algorithm to see the trend of these markers across the whole disease signature. And at the end, this was our study, and thank you all for listening. Thank you so much. So if you have any question, I'm happy to take this question.
literature. So are you using literature review, systematic literature review techniques, or another technique? And to provide accurate information related to the data. And for the second questions, for the project MENA, do you plan to provide more advanced marketing courses?
Yeah, thank you. Thank you for the question. So for the first question, so for the second question, for the second question, as I said, so in the frame of Bioinformatics, we did a lot in terms of building capacity. So in the frame of this project, so the main object is to continue to sustain all this and to continue to build the capacities by performing training, etc. Okay, so I give the example of IBT, which ran from many years and will continue with AfriGenD. And probably, so we have a working group, so we have a training sub-project here in the frame of AfriGenD that will work on, and we are continuing to think how to provide the different courses, either the same format like we did for Bioinformatics, but also a new format for in the frame of AfriGenD. So these training activities will continue in the frame of this project. For the first question.
It was for the SLR systematic.
Okay, for the curation. Okay, so for the curation, we are performing. So I showed you the example of AGMP, African Genomic Medicine Portal. So from the literature, we performed text mining of some abstract, but when we get data, so we performed many manual curation. Okay, which means that we have many curators that verify the data, and if something is missing or something is wrong, so they are reading the publication and extracted the correct information. Okay, so we have a round of cleaning of all the existing data that will provide for user. Okay, so it depends on the project, but mainly we are focusing on manual curation because we have many curators that are recruited in the frame of this project. So we are performing manual curation and different rounds or levels of manual curation. Okay. Okay.
For this presentation, so we'll take a question about the Bioinformatics Institute. If you could give us more information about it, or if you are planning to have some courses or workshops even with this Bioinformatics Institute. Thank you.
Thank you, Ghada, for this question. So the African Bioinformatics Institute is a great project or vision at the African level. So it will be, let's say, we will have a main hub, probably in South Africa, okay? But we need to have many institutions around Africa that will be involved in this African Bioinformatics Institute. And here at the Institut Pasteur, hopefully the Institut Pasteur of Tunis will be one of these institutions that will be involved in the frame of the ABI project. And we will probably have a meeting at the end of January in Cape Town. So in the frame of the ABI, and the meeting will be with the stakeholders to share with them the vision of the ABI and to try to get funds, because the main aim of ABI is to sustain the activities of Bioinformatics, AfriGenD, and so on. So now we have been able to secure some fund from Wellcome Trust and Chan Zuckerberg Foundation, but we need more because the vision is very huge, and it will not include 1, 2, or 3 institutions. It open to all institutions in Africa. So there is a link that have been shared to host to be a site within the African Bioinformatics Institute that you can find on the net. Okay. Thank you.
I have a question. Thank you, Mr. Hess, for your presentation. I have a question about, can you give us more information about the grant of Uganda? The grant of Uganda.
Well, Uganda is, so there is no grant, but there is a meeting which corresponds to the African Society of Human Genomics that will be held in Uganda in February from 3 to 7 February. So there will be a series of conferences related to to human genomics, especially, and in the frame of AfriGND. So we presented, so we applied for many sub-projects, and we have been successful. So we will present many projects during this this meeting in February. So it will be jointly done. So the African Society of Human Genomics, so the 15th anniversary, let's say, or event, but the first one of the Ugandan Society of Human Genomics, the first. So it will be joint and will be done in Uganda at NTB. Okay.
Thank you.
Thank you.
Thank you.
We start with the second session.
Thank you.
Thanks, Mr. President, for this nice presentation, and thank you for the audience for my question. Now we start the second session with the presentation of Dr. Mohammed. So Dr. Mohammed Hamd is professor of bioinformatics and computational biology at the JUC and the head of the Antivirus Analysis Group, Rostock University Medical. Dr. Mohammed does research in system biology, bioinformatics, and biostatistics. The presentation is entitled "A Basic Approach for Multiomics Integration." So, Dr. Mohammed, the floor is yours. Thank you.
Just, it's good. فريد accenito. I have some problem. Just a little problem for the microphone. It's okay. Okay, I can start. Just, I want to show this.
Hello again, everyone. Can you hear me well? All right. So today I'm going to talk about my scientific talk, which is a little bit different of the title, but so I have changed it in the last minute. So towards precision medicine and AI integrative approaches for multiomics data integration. So,
sorry.
Other problem?
Sorry. It's good. It's me. No. It's okay.
Okay. So back again. So today I'm going to show you some of my research team's work when we were in Rostock University Medical Center. We are located here, directly at the Baltic Sea, and I think Freya also was here as well. She was doing her research stay at the same building at IBIMA Institute in UMR. Well, so I'm going to start with this motivational slide again, which shows actually, the percentage of a patient population for which a particular drug in certain class is ineffective. And you see, for example, here the diabetes drugs, only 43% of population or diabetes population here respond positively to diabetes drugs, while the others don't respond. And here we know why, the old classical model or treatment model that's called one drug fits all is no longer valid. So the old classical model, of treatment or treating people was based on, assigning the whole group or all patients due to this certain disease to the same drug. And this tends to, or ends up with, like 3 different groups. The first group would respond positively. The second group wouldn't respond at all. And the third group, as you see here, would respond negatively, and it would end up with, adverse effects. Therefore, like almost a decade, one treatment model emerged here, and we called it personalized medicine. And this treatment model says, or based on stratifying patients based on their genomic variants or the genetic makeup, and we call this actually the biomarkers or prognostic biomarkers, where we are able to stratify patients into totally different groups. And based on the genomic makeup or the genetic makeup of those patients, we start to assign the most suitable drugs so that at the end of the day, all of them respond positively. So this approach started with the name personalized medicine, and later on was adapted to be precision medicine. So people, the whole community thought before that personalized medicine means to design or to develop a drug for every individual, which is not true. This is impossible, right? So that's why the community, later on made it better or named it better actually to be precision medicine. So we are treating groups of patients who have actually the same genetic makeup, and not only one specific individual or one specific, patient. That's why it was moved from personalized medicine into precision medicine. And here comes our role actually as bioinformaticians. So what's going to be our role here is to develop, to identify biomarker panels, accurate biomarker panels that allow us actually to stratify patients accurately so that all of them at the end of the day can be assigned to the most suitable drugs for them. And, that's our goal, as I said, but usually, we have got a lot of challenges. The first challenge is developing new drugs. So it takes actually many, or very, very long time, and also hundreds, thousands or millions actually of euros in order to develop new drug, passing from all phases of exploratory and the testing, and also the preclinical models and clinical models, and in order to get the final approval from the FDA until it gets also in the clinical market. Therefore, in my research group in Rostock University, we worked a lot on identifying synergetic drug compounds from existing drugs already in the market. The second challenge is the biomarkers, the prognostic biomarkers. Can we develop, like biomarker panels so that we can accurately stratify patients into those coherent groups? So right now, every approach for precision medicine in Germany or in Europe or wherever, so it depends only on the genetic mutations in some important genes. For example, in lung cancer patients, we basically focus on the KRAS mutations or the FGR mutations and so on. However, the genomics landscape is much more much more wider than this. So the idea of our research team here is to integrate all these different layers of genomics, like transcriptomics, epigenomics, proteomics, metabolomics, lipidomics, in order to identify, cooperative biomarkers from different and heterogeneous omics layer, so that we can reach at the end to an accurate stratification of our patients. So today, inshallah, I'm going to focus on, this area here. And if you are interested later on in the first part here, you can talk to me later offline. So our AI-based integrative approach is to integrate multi-omics data. So for example, why data integration is required for cancer studies here? So as you see here, so everyone is trying to resolve or to tackle the problem of the cancerogenesis based on the data set that he or she has in his mind, right? So, or in his data, in his hand, sorry. And then, so for example, the genetist tries to understand or to explain the problem of cancerogenesis based on the mutations and structural variations. The guy who has got transcriptomics data, so he tries to understand the problem or explain the cancerogenesis problem based on, the transcriptomic signature that he has got from his analysis, and so on. So all of them actually are incomplete. And the holistic approach wouldn't be really achieved until you couple all or gather all these pieces of puzzle together in order to have the holistic approach, of understanding the cancerogenesis process. Therefore, data integration is instrumental approach here in order to understand and to solve the problem of complex diseases. First of all, I'm going to give you one idea about two layers of radiological images, as well as also the transcriptomic layer. So here's an approach of integrating radiological images. So starting from large scale phenotypes or imaging phenotypes as we see here in radiological data sets, CT images, MRI images, X-ray images, and also trying to explain these large scale phenotypes under the representation of the underlying genetics here. So this work has been done by one or two actually persons of my research team. So Dr. Mohammed Tahoun, he was senior researcher in my research team in Rostock University Medical Center, and also Sarah Fisher, she is still a PhD student at Rostock University, and she is writing her PhD thesis right now. So in this research or in this study, they have implemented a pipeline, in order to to identify actually or to improve the prediction accuracy of prostate cancer patients. So if we talk about prostate cancer, so the clinical routines of prostate cancer, usually we start with the PSA test, prostate specific antigen test. So it's like a blood test. And then based on the certain level of this PSA antigen, you can judge whether this guy has prostate cancer or not, and also you can get some idea about the stage and the grade of this one. Second, we start also with the Gleason score. So at the next step, you can get a biopsy from the tumor, and then under the microscope, you can get the primary Gleason score and the secondary Gleason score. Third, by the digital rectal examination by the clinicians themselves, and also finally by the multiparametric magnetic resonance imaging or the MRI images. So all these clinical data have been gathered into some tables called pattern tables, and based on this information, the pattern table, clinicians can say a word about the correct grading on the pathological stage of the clinical stage. And based on that, the patient gets the corresponding treatment regime. However, after combining all this clinical information, the prediction accuracy of the prostate cancer rate reaches to maximum 80%. And now comes our roles as bioinformaticians. Can AI play a role here? So this is our research hypothesis. So we have got the clinical features here, like the age, like the Gleason score, the PSA levels, and our idea here is, can we add also biomarkers from genetics data like mRNA and also microRNA expression data in order to increase the prediction accuracy of prostate cancer patients from 80% probably reaching to very, very high accuracy. So that's our question, and my team there were able to develop what's called, PCA stage predictor. So the idea here, based on or starting from clinical data and also the genomics data, we start by data normalization, data preprocessing, and also identifying molecular characteristics of every prostate cancer stage here, starting from stage 1 till stage 4, and then we constructed the prostate cancer gene regulatory network, and further bioinformatics analysis, we were able to identify 4 potential biomarkers, 3 microRNAs, and one gene, as you see here. And then we incorporated all the genomic features as well as the clinical features here together into 3 different machine learning models in order to increase the prediction accuracy. And here we achieved a prediction accuracy of the pathological stage of prostate cancer patients up to 91%. So we were able to increase the prediction accuracy like 11 or 12% more. Furthermore, we looked also at the correlation between the large scale imaging phenotypes and as also the genomic features here that we can that we have acquired from the bioinformatics analysis here. So these are the imaging features of the 2 classes that we have been working on, so T3B and also T2C. And by doing simple correlation analysis, you see statistically significant correlations between the imaging features, as you see here, which are responsible for the aggressiveness of the tumor, and the genomic feature, the transcriptomic profiles of those 3 microRNAs and also the one gene. And you see here in both groups of T2C and T3B. So that was one study that has been already published successfully, and then we went further to make this pipeline automatic. How, starting from imaging data, as well as match it, and this is very, very, very important, otherwise we will not be able to say that we are discussing or analyzing multi-omics data. All data that you are working on must be for the same subjects, for the same patients, for the same sample that you are taking it. So this is called multi-omics studies. Yeah, so it has to be matched samples. Otherwise, this is not multi-omics analysis. Yeah, so the next study that we have gone for, it's a data-driven pipeline for RAM construction, radiogenomic association labs. So we would like to explain the large-scale omics or the large-scale imaging phenotypes here on lung cancer, within the light of the underlying genetics data. And here we expanded our genomics landscape into microRNA expressions, gene expressions, epigenetics, and also the clinical information here. So we started with imaging processing pipeline, so processing all these different image traits for lung carcinoma, and then we have used CT imaging and identified those features or the imaging phenotype that we would like to study. For example, the tumor volume, the tumor texture, the tumor size, the energy, the white-gray also features here. So those are the basic imaging phenotypes, and correlated this or associated this with the underlying genetics here. And this was a PhD work of one of my PhD students, Nicholas Spas, who successfully defended his PhD thesis 1 week ago, and also a part of Sarah Fisher's thesis as well. And the good thing here is they were able to build an automatic pipeline, so a data-driven pipeline that can automatically, capture these association relationships between the imaging phenotypes and also the underlying genetics. For instance, this is one example, and you can see here. So we have got two groups of patients. They have totally remarkable differences in their imaging profiles. Look at this. So clear difference between their, the tumor size, the tumor volume, the texture of the tumors themselves. Yeah, and by looking at the, the genetics makeup, so the transcriptomic profiles of their microRNA, as well as also the transcriptomic profiles of their mRNAs. So we have been able to identify those links between those two different cohorts of patients. And then we were able to relate these imaging phenotypes to the underlying transcriptomic signatures of microRNAs and mRNAs. We call this RAM, radiogenomic association map. So certain imaging phenotype is associated statistically significant with the underlying transcriptomic signature or genomic signature from the underlying genetics layers. And we can also visualize here which imaging features related to those genetic layers. So by doing this thousands times and replicating that on all data sets we have and in all biological correlates, we can finally end up with the RAM association for lung cancer. And this is not only applied on lung cancer, but can be also applied on other diseases, as long as you have the corresponding imaging data and the underlying genetics data. This is also another example where we have got also differences in their imaging phenotypes and also the identified or statistically significant microRNAs and also the genes. Then we can also build what's so-called the gene regulatory network from those microRNAs or the related or associated microRNAs and genes, and build using the TFMIR website. So we have now the version 3 of TFMIR in order to identify those interactions between the microRNAs and the genes. Then looking at one hub gene here, for example, and reflecting this on single-cell genomics data from Lamprecht et al. This is one single-cell genomics data set that's quite famous for lung cancer. Then we can know exactly those hubs, where they are expressed, which cells are expressing those hubs, so that we know also what kind of cells that could be responsible for having these differences between those imaging phenotypes on those patients and those patients. So you see now we started very large, so imaging phenotypes from imaging data, and then we started to nail them down to relate them into the corresponding transcriptomic signature or associated with certain transcriptomic signatures. And furtherly, by doing single-cell analysis, also we're able to get to know some insights about what cellular cells or what cells could be also potentially responsible for this kind of differences between them. So starting from larger scale imaging data and ending up with single-cell resolutions of the relationship. Then we moved also afterwards to from bulk transcriptomics to single-cell transcriptomics, and this is some research I would like to give you about what I'm doing in my research lab, also in Rostock University, as well as the German University in Cairo. So we all know that the main differences between bulk transcriptomics data, so we take a biopsy from the tumor, for example, and start to extract the genetic material. However, the genetic material is kind of averaging or, averaging for all populations of cells. It's not really pure like this one. So this would be representing the bulk transcriptomics. In contrast to this, if we use certain technologies, especially the 10-axis genomics technologies, so we can basically get the transcriptomic profile of every individual single cell. Here. So this could help basically in building one, what's so-called single cell atlases. And here is an example of one immune atlas we have developed, specifically from 28 integrated data sets from 20 cancer types and from 500, more than 550 donors. As you see here, so we have collected almost more than 1.25 million cells from different data sets in order to give us more confidence about the expressions of certain targets, the communications between the cells and each others, if we would like to study any kind of cancer entities. Then we are also moving to the area of spatial transcriptomics, and spatial transcriptomics here is adding a new dimension on the single cell data. So right now, we have no idea about the spatial information about these cells. So we know that we have got different cell population, different cell compartments. However, we don't know basically the spatial resolution, the spatial information, where they are located. The proximity relationships between these cells and each other. And in some applications like the tumor or the tumor microenvironment or the immunotherapy, this kind of information will be much more important to know than also the genetics as well. So for this, we are adding also the spatial information here in order to identify, for example, the spatially variable genes. So the same cells, for example, they are having the same annotation. So both of them are, let's say, B cells or T cells. However, one population is quite close to the tumor cells, trying to nail them and kill them down. While the other cell type, it's not actually active. So they are not infiltrated towards the cancer cells. And we would like to know why. So by doing simple analysis, simple bioinformatics analysis, we could identify why these T cells are inactive and they are not infiltrated towards the cancer cells. And basically we can boost that, and we can, like using certain therapy, we can boost the immune cells towards the cancer cells. So the second last example that I would like to talk about, it's also one research project that's going now on in my research team at the German University in Cairo. So the hyperspectral images versus the spatial transcriptomics data. So this is an ongoing project between my team and also Dr. Salim's team. So Dr. Salim, he is an expert in developing machine learning approaches and deep learning approaches on hyperspectral images. And in hyperspectral images, we have the spatial information and also a vector of wavelengths for every urban entity. So we have customized an in-house developed pipeline, deep learning pipeline by Dr. Salim and his team on spatial transcriptomics data. So imitating, the spatial transcriptomics imaging with the hyperspectral images. So the vector of wavelengths here would be actually the single cell profile of every cell in order to deeply characterize and annotate the different cell types. So this project is led by Mariam Wael. So she is a master's student at the German University in Cairo. So another project also is going on. It's all about the multimodal fusion of omics data. This is also AI-based using deep learning approaches. And as you know, we all humans, so we have capabilities to learn from different senses, like we learn also visually from our ears, by reading, by writing. So utilizing all the things that we have in order to learn from different sources, because we are humans, we have been created to learn that way. However, for computers, no, it's totally different. Therefore, we have to develop deep neural network approaches in order to teach the computer how to learn from different sources, how to learn from different and heterogeneous omics data sets using artificial intelligence approaches. And this is basically has been inspired by these publications, cancer cell publications, when they integrated, histological slices together with copy number variations of genomics data based on copy number variations and mutation landscape in order to accurately predict what's called the C index, the survival rate. So the C index is kind of a score that's indicative to the survival rates of those patients. So as you see here, when combining two omics layers, so think about the last one here, so the blue one, yeah, usually you achieve a better C index, which is, as I said, indicator for better survival rate prediction. So inspired by this, Ala Amr, she is also a master student in my research lab at the German University in Cairo, is developing hyper deep learning models, and we mean by hyper that we also integrate deep learning approaches together with the classical machine learning approaches. Both approaches are in the same pipeline, so we learn by the deep learning the most important features, and then applying this into classical machine learning as well. So here we use three different omics layers. So the genetic information here represented by the copy number variations and transcriptomic signatures, RNA data, and radiological images, as well as also the pathological images that we get as well. And she is now doing her research stay at Göttingen University. She has got also extra fund from the GARN project in order to continue working on this project at Göttingen University in Germany. So I hope I was able to show you a couple of examples of integrating multi-omics data sets. However, it's the way is really long. So we have got a lot to do in order to be able to integrate also all other omics layers, including the microbiome, the epigenome, and proteome, and all these different and heterogeneous data sets, if we would like to have a reliable precision medicine system to rely on. With this, I would like to thank all people behind my research who funded me. So basically the DAD, so a couple of funding rounds, as well as also Rostock University and the Forum Grant also from Saarland University in Germany. And I would like to thank also all people behind this work. So starting from Mariam, Ala, and Nicholas, and Sarah, and also recently Dr. Mohammed Nagi, he is also a senior postdoc in my research group. And I would like to thank you for your attention. Thank you.
Thank you very much, Dr. Mohammed, for this nice presentation. Now the floor is open for the audience for a question. Okay.
Thank you for this nice presentation and continue the great work. So I have basically two questions. The first is you said the differentiation between precision medicine and personalized medicine. So are you with the strategy of elimination of the term of personalized medicine?
Yeah, it's confusing many people, I would say. So I would prefer to use always the precision medicine term instead of personalized medicine and so on, because personalized means like you are personalizing it for one single individual, and this caused a lot of, this is practically not possible. You will not be able to develop and spend a lot of money, bunch of money, just for one person. No, it doesn't work like this at all.
But that's still very important for certain cases.
Yeah, but we are talking about global, term to be, generalized later on. So that's why people are moving towards precision now, not really personalized anymore.
So the second question is about the data that are using this different.
Yeah.
Are they public or connected?
Yeah, that's a very good question too. So, in order, we are retrieving or identifying patterns, right? So in order to do this in the right way, so you have to have training data and also validation data. So training cohort and validation cohort. So we usually use TCGA cohorts or the American cohorts for training our models because it's really huge data. And then we are using also different cohorts coming from the ICGC, for instance, or our proprietary data, so our private data sets that we have also in Rostock or in Göttingen, to validate the patterns that we have got here on there, right? So we have to use this way. Otherwise, it will be just like, could be for coincidence. Okay.
Thank you so much for this presentation. That was very interesting talk. My question is a little bit technical. I want to know the integration method or technique you use for this type of data. And in general, how do you choose the appropriate integration method?
What? All right. So I have explored like 4 projects. Okay. So the first one by Sarah and Nicholas, and those ones, you know, they have used basically like the very naive approach, which is, you know, getting or identifying the most important features from every individual omics dataset, and then simply integrating them into one machine learning model, and then getting the prediction results. So that was one idea. The second one, which is, you know, I mean, the one that Ala is working on it right now, there are many like different approaches for fusing the datasets. So one is basically using totally deep learning approaches. The second one basically using not statistically, but classical machine learning based. And the third one that we have been using here is the hybrid model. So using the deep learning approaches and also the machine learning, classical machine learning approaches. So based on our results, and we have tried all actually, but based on our results, we have got, you know, the best results on the hybrid model. That's why we adopted it here in this approach.
Thank you so much. Thank you, Dr. Mohammed, for your great presentation. I have a question. Could you explain more or elaborate more the idea of integrating single cell analysis and spatial transcriptomics in the area of precision medicine?
Okay. That's really, you know, a very future question, I would say. So, yeah, the precision medicine and the incorporation of the spatial transcriptomics data in precision medicine. So I would say till now, all research that has been done, you know, in spatial transcriptomics data and single cell genomics wasn't really be like a practical, or didn't give you more insights about the practicality of applying it in precision medicine or in any clinical routine. So precision medicine, at the end of the day, it's a clinical routine. You would like to stratify patients and then assign them to the best treatment, right? And this is why, because the extra costs, it's really costly. So one sample costs, for example, one sample for, you know, single cell transcriptomics and spatial transcriptomics could cost up to 6,000 euros. And you can imagine also whatever you do in Krankenkasse or the medical insurance companies, for example, even in Germany, wouldn't accept to pay or to afford 6,000 euros just to profile, you know, one patient and at a certain time. Imagine we are doing these approaches, you know, based on time series data. So we would like to get this profiling on different times, you know, during the progression of the cancer. So it wouldn't be practical because of, you know, the extra cost. Some trials, I know a couple of research institutes, I think that was in Broad Institutes in America. So they are trying now to, you know, like make use of this technology by searching for, you know, better and even cheaper approaches for, you know, having or doing this spatial transcriptomics data for the sake of precision medicine. But right now, the application in terms of, you know, the practicality still, I wouldn't say, wouldn't be possible in these days. I would expect in 5 years or 10 years, we could expect this, like what we have been talking now. Like 10 years ago, we've been talking about the NGS profiling. It's almost impossible to afford it. Right now, you can do it with $200, right? But right now, it's not possible, I would say, in the future.
Okay, thank you. Thank you, Dr. Mohammed, for this explanation and addressing work. So for the first project that you are working on, prostate cancer, you mentioned that you've chosen some machine learning model to classify the stage of prostate cancer of the patients. So how you've chosen the upper
model of machine learning, and did you base just on accuracy of each model to choose between them or not?
All right. So for classical machine learning, so we have tried to, to try all possible classes of classical machine learning models. For example, we have used random forest as an example for decision-based classification. We have been using also logistic regression model also for classification. So it's based on, like a linear classification as well in the logistic space. And we have been also using the Bayesian naive classifier. So basically, like trying different classes of machine learning. And I can say here that the one that works best or outperformed here was the decision-based one, the random forest. And this is not also a coincidence. So, random forest always, not always, most likely outperforms, specifically in classification tasks, and also when you have got, omics data. Yeah.
Thank you for your response. I have a little bit of suggestion. So I worked with leukemia cancer, and I've tested, but in the final I found the model that goes to classify the test maybe from 10 to 20 model, and give you an idea about the best model that fits with this stage of patients, and then you can build on this in every stage. Choose the appropriate model from this model classifier.
Yeah. Okay. Thank you. Shema has got a question.
Thank you, Dr. Mohammed, for this excellent presentation. I just have a question, technical question. So when you said for the machine learning that you use the different models, predictive models, until you find the best fitting one. The problem is, have you had any problem with overfitting of your data? And if how large the data set that you work with, and how do you tackle this overfitting issue?
All right. So basically, we have got a lot of these problems. So anyone who works in machine learning definitely will get some times to treat with overfitting data or overfitting models or underfitting models. Yes, we have got that many times. And this is, if we do this, for example, if we get something like this, if we, by using, for example, linear regression models or linear classifiers, so we are using more complicated models in order to, for overfitting, we are using a little bit simplifying models, so the other way around, sorry. Yeah. And also trying to, increase our data sets in order to have, like better data, and using as well the cross-validation. So cross-validation has been originally, developed in order to minimize the overfitting approaches. So yeah, my team also has encountered a lot of these things, and you can simply, treat with that using either cross-validation, and you can check also the best K based on, like the evaluating later on the training model and the testing model, and using also simpler models, right? So by using simpler models, so you can minimize the idea of the potential of having, overfitting. Yeah, the data set, we didn't have a problem in the data set size at all. So TCGA, we have got a lot, hundreds of patients, hundreds of cohorts. Yeah, for the ICGC, yeah. Probably for the testing data, yes, because testing data, as I said, we have been doing this on proprietary data, private data sets. So I think it was maximum of 100 observation of 100 samples. So which is also reasonable as a test data. But for the training, no, we didn't find any problem in the size of our data sets. And one thing, why did we choose also lung cancer data and lung cancer patient to visualize this? Because lung cancer and breast cancer, they have got thousands of samples there. So in comparison to other cancer entities like head and neck and prostate and so on.
So, thank you. But let's say if we did the cross-validation and everything, like we did everything spicy, but we still have an overfitting issue. Let's say, how do you between training and validation set, how many the gap that you take into consideration between the accuracies of the training and the validation set to say, okay, I'm an overfitting? Is it like 5%, 10%?
So it's not like this. We are not using about certain number of percentage or gap. So we usually plot the training error versus the complexity of the model, and also the testing error. Also, again, is the complexity of the model. And then we see, the intersection point that could be, okay, acceptable here and acceptable there. Yeah, and this point basically determines that I wouldn't say the best, but I would say the optimal, the optimal complexity level of that model. Yeah, if all of that didn't work out, then that means that you have either a problem in the selected feature that you are implementing on, or also you have a problem in processing your data sets. For example, you are doing, for example, the normalization before or after the splitting of the data, so you have allowed some sort of, information leakage in the training data. So usually there are some tips and hence here. I think Dr. Muhammad Salim and I planned something for tomorrow in the, he's not here, okay. So tomorrow we'll have a workshop for the best practice of machine learning. So we will be giving also or highlighting the best tips and tricks here to have a machine learning model based or running on transcriptomics data as an example of the workshop tomorrow. So be there.
Thank you.
Okay.
Thank you for your great talk. It's great to have you welcome back to IDT. So my question is regarding the single slide analysis. So did you focus only on analyzing the cells that are from the tumor tissue, or are you also investigating peripheral blood cells, especially the immune cells? Because we know one of the let's say the main objective is trying to find early biomarkers for any disease, so to help prevent the aggressivity of the disease and to have like better management of the disease.
Yeah. Well, in the example I showed you here, so we were focusing on immune cells only. So building an atlas, usually you have to know your target. What are you trying to do? Yeah. So this atlas has been developed in order to support, the development of immunotherapy drugs. So specifically in IO drugs, immuno-oncology drugs. Therefore, we would like to focus only on the immune cells. That's why it's called single-cell immune atlas. If we, for example, if I'm working with Dr. Nabila, for example, she's working on neurodegenerative diseases, they will be focusing basically on certain kinds of cells, right? So the brain, for example, or certain region, right, in the brain that's responsible for developing this disease that she is working on, right? So it should be target-based. You know exactly why you would like to develop this atlas, and based on that, you decide what kind of cells should be involved and should be, integrated and pulled from the different data sets in order to build this single cell atlas. Yeah, so that's the idea. It's case-based.
No more questions. Thank you very much.
We can take it offline. Okay.
Thank you so much for this nice presentation. It's very interesting, but I think a lot of us have more questions, but we will have time to have tomorrow and after tomorrow to make questions. Thank you so much.
All right. Thank you so much. Bye-bye.
And then I will go to give us. Okay, so we will. Go to the note. Okay, so we invite Dr. Maria Henry to provide us a short exploration of genomics and bioinformatics.
address the same terms for ALS. Is currently professor of molecular medicine and pathology at the German University of Cairo and the lead of the molecular pathology unit. Dr. Hani graduated from the Medical University of Sfax. She is Tunisian and spent the practical years of her medical studies in Munich, Germany. She received her PhD in molecular genetics pathology in 2009 from the University of German University of Cairo. So, welcome, Professor Ghedira, and the floor is yours. Thank you.
Thank you.
I will ask people to don't give more than one question because we are so late, and Dr. Ghedira and other presenters will be here with us, and we will have time to ask questions.
اسمع الماوس هذه تظهر الماوس هذه كنحطها كتظهر له هنا لا هذه كتظهر له تظهرش رجعها كيف تمشي
So, good afternoon. It's my pleasure to be with you for this fourth bioinformatics workshop. And first of all, I would like to thank my dear colleagues from Institut Pasteur de Tunis, Dr. Qais, Dr. Osama, Dr. Reem, and their team for organizing this nice event. I will be talking about one of our disease of interest in our research group, which is amyotrophic lateral sclerosis. And last year, I tackled mainly the microRNA sequencing study that we did for this disease. And during this workshop, I will be mainly tackling the genetic aspects of ALS. So ALS stands for amyotrophic lateral sclerosis. It was founded in the 80s already by the French neurologist Jean-Martin Charcot, but it was rather more known among people when a famous football player named Lou Gehrig was affected by this disease. And that's why it is also called a Charcot disease or a Lou Gehrig's disease. This ALS is a very complex disease, and in Egypt, it was not quite known until 2020, when a famous football player, Mohamed Zakaria, was affected by this disease. He started to have the symptoms in 2018, and in 2020, he was already on wheelchair. So it is very fast progression. And the interesting point is that he was very young, which was not usual for ALS. So we heard about the challenges in tackling complex diseases and rare diseases, and ALS is a prototype actually for complex diseases for several reasons. First of all, it's a rare disease, and we heard today in the morning the debate about the rare disease. Although the definitions, there is no consensus definition regarding rare diseases. We have the European version and the American version.
So in general, its prevalence is 3 over 100,000, which is rare. However, we don't have epidemiological studies or incidence and prevalence data from African countries, and there is an expected increase in the number of ALS cases in our continent. Another aspect of this complexity of the disease is that it is very heterogeneous on the clinical level. Regarding the phenotypic heterogeneity, it has a very complex and mysterious pathophysiology. It is a devastating disease that leads to the death within only 2 to 5 years for the majority of patients. It is a multi-system degeneration, not only the motor neurons, and we see a great interpopulation variability on the clinical level, phenotypic level, genetic level, and also the epidemiological aspects of the disease. So ALS is a motor neuron disease, is a part of the neuromuscular diseases, and it affects mainly the motor neurons. It can affect the upper motor neurons or the lower motor neurons, which leads to weakness of the muscles and ultimately paralysis. In addition, it is characterized by the progression of the disease from one muscle groups into other muscle groups until it affects the respiratory muscles, and this is the cause of the disease within, as I mentioned, 2 to 5 years for the majority of patients. Regarding the pathophysiology of ALS, it is quite complex, and I will be mainly talking about the genetic aspects of this disease. And if you look at this graph, you can see that many cellular processes could play a role in the pathophysiology of the disease, ranging from hyperexcitability to mitochondrial stress, oxidative stress, axonopathy, impaired protein homeostasis, nuclear transport, and similar to the other neurodegenerative diseases, we see again the complexity of the pathophysiology that is still mysterious and not fully understood, especially that we have many genes that have been associated with the disease. We are not talking about a single gene, but many possible genes. And you can imagine for each gene, you can have several mechanistic roles. Some of them, they are known, and many others, they are still under investigation. And I will dedicate my talk to the ALS genetics. And here you can see that we have many genes that have been associated to the disease, mainly by studying the familial cases, because we have 10% of the ALS cases, they are familial. This means they have family history and other members of the families are affected, while the majority, they are sporadic without any family history. So what do we know about ALS genetics? We know that there are many genes. You see them here. The first gene that was implicated in ALS was associated with the disease is the SOD1, superoxide dismutase 1, which is the focus of in my talk. It was discovered in 1993. And with the completion of the Human Genome Project, you see the exponential increase in the number of genes that have been associated with the disease. Regarding their implication, they have been mainly implicated in 3 pathways or mechanisms, mainly protein homeostasis, RNA binding proteins, and cytoskeletal proteins. And if we focus on the SOD1, which was the first gene associated with the disease, we know SOD1 is the superoxide dismutase, which is an important antioxidant enzyme. It contributes or it is crucial for the neutralization of the superoxide anion, which is a very active free radical, by converting it into the molecular oxygen and hydrogen peroxide. It is a metalloenzyme, so it requires copper and zinc. Copper is crucial for its catalytic activity, while zinc is mainly important for the stability of the protein. Regarding its expression, it is abundant and highly conserved, mainly cytosolic enzyme, but it can be found in the mitochondria and the plasma reticulum. It makes up roughly 1 to 2% of the total soluble proteins of the CNS. And as I mentioned, it requires copper and zinc for its activity. So this slide is to show you the gene structure as well as the protein structure of SOD1. For the gene structure, it's composed of 5 exons. It's relatively small protein, 5 exons. It is associated with the family ALS, roughly 12 to 23% of cases, familial cases of ALS, and 2 to 6% of all cases, both familial and sporadic ALS. It is mainly for the familial cases inherited in autosomal dominant manner, although we found also autosomal recessive inheritance. Most of the mutations, they are missense mutations, and some deletions or frameshift mutations, they have been also found. Regarding the protein structure, it is a homodimer. It is composed of 2 units, 16 kilodalton each subunit, and 153 amino acid. It's a copper zinc superoxide dismutase. The units or the subunits, they are connected by a disulfide bond, and it has a beta barrel core structure and stabilized by a disulfide bridge and a zinc ion in the active site. So what makes SOD1 really challenging to study is the number of possible mutations that have been found in the gene that have been associated with the ALS disease. And we have more than 200 mutations have been now reported, and you can find them in the ALS database. And you can see here the 5 exons in the blue color, and you can see that the mutations, they are spread literally throughout the exons. There is no hotspot region in the gene, but all the exons, the 4 exons, could be affected. And when you look at the distribution of the mutations, you can find some of them, they are found in the dimer interface, others in the zinc binding residues, other mutations in the copper binding residues, or in the cysteine residues forming the intrasubunit disulfide bond. And that makes actually the functional studies of the mutations challenging. And you will feel the debate that is now happening regarding the SOD1 mutations, and you will know why is SOD1 now gaining more and more interest for ALS patients. So based on its role as an antioxidant enzyme, the first hypothesis that was suggested how the SOD1 contributes to the disease was through a loss of function hypothesis. Why? Because we know that oxidative stress is a very well established pathophysiological mechanism in many neurodegenerative diseases, and ALS is one of them. Second reason was the discovery of disease-causing mutations in the SOD1 gene in the familial cases, and the finding that free radical damage was found in the CSF, serum, and urine, even in post-mortem materials from ALS patients. And this was the first hypothesis in 1993 that SOD1 could lead to ALS through a loss of function mutation. However, a couple of years later, with more research, the hypothesis of loss of function was abundant, and they switched to gain of function hypothesis based on several aspects. First of all, they found that in humans, there was no correlation between the SOD1 dismutase activity, enzyme activity as an antioxidant, and the aggressiveness of the clinical phenotype. And then they did several interesting studies on mouse models, and when they analyzed the SOD1 knockout mouse model, they found that these mice, they didn't develop the typical ALS phenotype. And when they they developed transgenic mouse models for SOD1 mutations, and there are now several transgenic mouse models for SOD1, these transgenic mouse models, they actually characterized by an increased dismutase activity. Why? Because they have their own SOD1 activity, and then we express the SOD1 mutated SOD1 activity. So, and they found out that these mice, they, despite the increased dismutase activity, they develop an adult onset motor phenotype with distal, with affection of the lower motor neurons, which goes along with ALS. So this means that the mechanism is most probably not a loss of function, but it is most probably a gain of function and a gain of toxic function. And the other observation is that when they developed transgenic SOD1 for a certain mutation on a normal mouse background, this means they have the wild type, and they compared the survival time with the mouse, transgenic mice expressing the same mutation, however, with knocked out SOD wild type. they found no change in the survival time. And all these research studies, they supported at that time the SOD1 gain-of-function hypothesis, that is most probably through the aggregation of this mutated SOD1, and these aggregates, they have like a toxic impact and toxic function on the neurons, and this leads to the degeneration of the motor neurons. Especially that SOD1 aggregates were found in the spinal motor neurons in histopathology, in SOD1 familial cases, and it was research was trying to understand how these aggregates could lead to the ALS pathology. In addition to the aggregation in the cytoplasm, these aggregates, they can migrate from the intracellular into the extracellular environment, and they can activate the inflammatory cells in the brain, leading to microgliosis. In addition, the SOD1 mutated forms, they were found in the mitochondria, leading to mitochondrial dysfunction and endoplasmic reticulum stress. So based on this toxic gain of function, there was the initiative to target the SOD1 as a potential therapeutic target, and the idea was to silence the SOD1 gene, the mRNA gene, using antisense oligonucleotides to prevent the formation of these toxic aggregates of proteins. And this was the approach that was studied, and in 2006, the first studies on transgenic SOD1 mouse models, or rats, they found that antisense oligonucleotides were effective, and a phase 1 trial was already completed in 2012, showing that low dose of SOD1 antisense oligonucleotides showed no adverse effects compared to the placebo. And this is to show you the mechanism of the antisense oligonucleotides, which are short sequences that will bind to the mRNA of the SOD1 and prevent the translation, and thus preventing the production of this aggregated protein and the toxic effect on the neurons. So, and with these trials, the first trial was published 2013, and at this time there was the active groups and the experts in the ALS research, they were very quite interested in studying more the SOD1, and to reconsider the loss of function and the role of the SOD1 loss of function in amyotrophic lateral sclerosis, knowing that these antisense oligonucleotides, they will target the SOD1, not only the mutated, but also the wild type. So there is a risk of significant decrease of SOD1 in the patient receiving this therapy. So they reconsidered, they did a very nice review published in Brain in 2013, summarizing the experience of 2 decades of research in the ALS field, and trying to highlight the implications, the possible implications of SOD1 loss of function for current therapeutic approaches. And they summarized, or they collected the data regarding the different mutations in the SOD1 gene, regarding their enzymatic activity. And you can see here in the graph, these bars, gray bars, they represent the percent of enzymatic activity as percentage of the normal activity in different patients having various mutations, okay? And you can see that the wild type, this is the first bar on the left, it is 100% enzyme activity, and you can see that the majority of the mutations, they show a decrease in enzymatic activity, roughly reduced to half of the normal enzymatic activity. They found also that homozygous SOD1 mutations, they have different activities ranging between 25%, like the last one on the right. This is homozygous mutation with only 25% enzymatic activity, and compared to the fourth column from the right, which is homozygous, another homozygous mutation with almost 100% enzyme activity. So basically, we can classify the SOD1 mutations into wild type-like. This means they preserve the enzyme activity like the wild type SOD1, and other mutations, we call them the metal region binding mutations that will decrease the activity of the enzymatic activity of this of this enzyme. So you can see that it is quite difficult to understand the functional impact of each mutation regarding its enzymatic activity. And we didn't know in 2013 when this review was published, we didn't know the impact of a complete loss of SOD1 enzymatic activity on humans. We have data from the transgenic mice, however, they cannot be extrapolated to the humans, and that's why there was a lack of a human model where you have 100% or complete absence of the SOD1 enzymatic activity in order to assess its pathological impact. However, in 2019, and this was another breakthrough publication published in Brain. They reported for the first time about a human SOD1 loss of function phenotype. So this was a family with consanguinity, and the child, was affected with a severe phenotype, neurological phenotype. It was different from the ALS, but it is still a neurological phenotype with a more generalized neuronal phenotype in children. And this was due to this homozygous truncated variant of SOD1 that resulted in total absence of the SOD1 enzyme. We see the wild type in the first bar on the left, followed by the parents, heterozygous parents, and then the last bar, you see the complete absence of SOD1. And this is actually a proof that the complete loss of SOD1 enzyme is actually pathologic. And it has to be taken into consideration when we think about silencing the SOD1 gene through antisense oligonucleotides. And this was actually the main message of these studies. So the clinical trials, of course, using SOD1 antisense oligonucleotides, they continued, and the phase 3 Valor and its open label extension was announced actually in June 22. And this drug, antisense oligonucleotide, received the accelerated FDA approval in April 2023, so it was last year. It's called tofersen or Qalsody, and it is for the treatment of SOD1 ALS patients. It is only for a certain percentage of SOD1 of ALS patients who have SOD1 mutations, and this was actually the first gene-based therapies for ALS, and it is actually a therapeutic breakthrough. Yet we need to think about possible implications of SOD1 loss of function for these current therapeutic approaches. So now I would like to share with you our experience with the field of ALS genetics, and our approach was to screen for the first time for familial and sporadic ALS cases, patients in Egypt, and we adopted a panel analysis, a whole exome with panel analysis comprising 35 ALS genes. For the sporadic ALS patients, we couldn't find ALS-associated mutations, but for the familial cohort, it was a small cohort, 11 families. However, we found interesting results. And this was the first report into about the first insights into the genotype and phenotype of familial ALS cases in Egypt. And this was characterized with an early onset and high rate of consanguinity as expected. The Tunisian group, they published before us the first genetic data about ALS roughly 2 years before us, and it is already published. So our approach in studying the pathogenicity of the mutations of the variants was the classic pipeline. We used several bioinformatic databases to study the allele frequency, the documentation of the variants in the different databases, the functional impact, computational in silico impact, and so on. And this is a summary of our mutations that we found in the Egyptian patients. So you can see the high rate of consanguinity in the majority of the families, and the presence of homozygous mutations, knowing that homozygous mutations in ALS patients are very rare. And the most striking finding is the high frequency of SOD1 in the Egyptian population. So among 11 families, we found 4 families having SOD1 mutations. SOD1 mutations is the second or the third mutation, common mutation in Europe. It is number 1 in China, and it is number 1 in Egypt, obviously. And interestingly enough, in Tunisia, it is number 3. It's rank 3. So in Tunisia, we have the TAR-DDP, C9orf, and then comes the SOD1, and it shows that we have a great genetic variability even on the African continent. Maybe we'll have our database regarding the SOD1 mutations across Africa, because there are now more active groups studying the genetics in Africa as genetics. We have our group in Egypt, we have the group in Tunisia, group of Professor Ghedira, and South Africa, they are also active in studying this disease. So, and the highlight of this study was the SOD1, and I focus on the SOD1 mutations. I don't know whether it is clear. See, it looks hazy here. Among the 4 SOD1 mutations that we found, one is particularly interesting, and this is a homozygous mutation. As I mentioned, homozygous mutations are really rare. I think worldwide we have only 10 to 12 homozygous mutations. I cannot click the screen. Any help? And then? Arrêtez partager. Okay. Yes, yes. Let's go. So. Yes. So this homozygous mutation is particularly interesting, and it was published as a case report in Journal of Neurology because it was not previously, we couldn't find this mutation in the databases, so we called it novel, okay, with caution. It was not reported. It is an exon 3, which is the exon least affected with the mutations in comparison to the other exons. It is close to the zinc binding site. It was found in a family. So this is the pedigree of the family. The parents were first cousins, and the arrow shows the patient who developed, a female patient who developed the disease at the age of 26, really young. She was even pregnant. She developed a foot drop with very rapid progression and bulbar involvement. This means involvement of or weakness of the bulbar regions, and it was very rapid progression, and she died within a few months. The family history showed that the patient had a younger brother, and he developed a similar phenotype at the age of 10 years. He was really a child, and he was misdiagnosed, of course, because no one could think about ALS in a child. And then he was diagnosed 2 years later with ALS, juvenile form of ALS with rapid progression. No genetic testing was done for her brother, but we did genetic testing for the family, for the patient, parents, and the unaffected brother. And we found this homozygous mutation in the patient. This both parents and the brother were carrying the heterozygous form of this mutation. What was interesting about this family, we pushed the investigations a little bit. We measured the biomarker neurofilament light chain, which is a biomarker for neurodegeneration, and we found it very high. So the cutoff is 45 picogram per milliliter, and the patient had 148. So this is very high, and in comparison to the heterozygous family members. And most interestingly, we could measure the enzymatic activity in the RBCs of this patient. And for this, we collaborated with a specialized group, specialized and expert in measuring the SOD1 enzymatic activity in RBCs, because it is difficult, challenging, and we have problem of reproducibility. So we found 16% only of enzymatic activity, and this was the lowest. This was a record, actually, and this was the lowest enzymatic activity ever reported in ALS patients. The lowest previously known was 25%, and we had 16% in our patient, with much higher, of course, in the heterozygous unaffected relatives. And this publication was actually confirms actually the importance of studying the enzymatic activity of the different mutations and to think about the impact, the functional impact. So we have here this patient with a very low, 16% only of enzymatic activity, very severe clinical phenotype, and death within a very short time. So we were interested now in studying the functional impact of our mutations. So we have 4 mutations, SOD1 mutations found in our familial patients. One of them is homozygous and novel. The second one is novel in terms of the missense mutation, and the 2 others, they were recently published as pathogenic or likely pathogenic mutations. So our approach here is to study the aggregation potential of the SOD1 mutations and the enzymatic activity. So both will decide about the pathogenic role of the SOD1 mutations, and our approach is to use a fluorescence microscopy and Western blot and some co-aggregation assays to measure the aggregation and to measure the enzyme activity through an activity assay developed by our collaborators in Germany, and also gel assay. So what we did is we designed primers for our mutations, and we wanted to compare the Egyptian mutations to the European or German mutations to compare this interpopulation variability. We induced mutagenesis using overlap extension PCR. We ligated these mutants into plasmids, DNA plasmids, and we transfected the constructs into hex cells, and then we used the cells and the lysates to conduct our experiments. This is an ongoing project. It's a joint project between our university and Ulm University in Germany. So these are just preliminary results, but I wanted to share them with you because I thought it fits well in the theme of this workshop. So these are images from the fluorescence microscopy. I don't know whether we can see clearly the colors. So the first row represents the wild type. We have the DAPI staining. The blue one represents the nucleus, and the red fluorescence represents the cytoplasm. So the blue mutation is one of our Egyptian mutations, and the red one, G93A, is widely reported mutation in Europe, and we can see here, but you have to trust me that these arrows, they highlight some aggregated SOD1 inside the cytoplasm. We did some Western blot studies here to compare the aggregation potential of the different mutations. So as I mentioned, we are working on 4 Egyptian mutations in the blue color, and 2 other mutations. The choice of the 2 other mutations, the G93A, is widely common in Europe, and it has a normal enzymatic activity. And the second one, the G85R, has a very reduced enzymatic activity. So we are analyzing our results to these 2 mutations as references. And the first on the left, Western blot, is under denaturing conditions, and you can see that we have the monomers. We can see the monomers in all types of mutations. On the right side, this is non-denaturing conditions in order to visualize the aggregated proteins. And you can see that the wild type on the left, it doesn't show any aggregates, only the monomer. And then we have the majority of these mutations, they show this smear. And this smear is actually, they represent aggregates, aggregated protein. Interestingly enough, the G38E, one of our mutations, it's very similar to the wild type. We don't see any aggregates here. And our homozygous mutation, novel mutation on the right, the S69P, we show less aggregation and only a few dimers and the monomers. So we can clearly here see that we have differences in the aggregation potential of the different mutations. And if we translate these Western blot staining into numbers, you can see that the wild type is composed of 100% monomers and no dimers or no aggregates. However, the S69P, the last one, it has only roughly 6% aggregation if you compare it to the other mutations. And the G38E is also interesting because it doesn't contain any aggregation, and it has the same aggregation or the same pattern like the wild type, and we see only monomers. And here again, if we focus on the S69P, the novel mutation, we might think of returning back to the loss of function hypothesis because it has a very low enzymatic activity. It is associated with a very severe phenotype. And when we look at the aggregation, it doesn't really aggregate like other mutations. So we can discuss that we see here it could be a loss of function rather than a toxic gain of function. We are working further on this project. We need to do the enzymatic activity gel assay and the enzymatic activity assay in order to confirm our results. And these genetic studies that we have conducted, it is only addressed to a certain percentage of the ALS patients, because the other patients, they do have other mutations, or the sporadic patients, they didn't show any ALS associated mutations. And that's why our approach is more holistic. And our holistic approach in ALS, we are integrating several omics layers. On the genomics level, we have a PhD student who is working on further analyzing the genomic data on the exome and the genome level, and focusing not only on the non-synonymous, but also on the synonymous variants. For the epigenomics, we have already tackled the microRNA sequencing for ALS, and we are now working on the methylation, the methylation of these patients. Metabolomics, we have Mariam is doing her PhD and tackling the metabolomic profile for ALS patients, and our plan is to integrate several omics layers, okay, and this is supported, this collaboration with my colleague, Dr. Mohammed Hamid, to integrate the genomic data, epigenomic data, metabolomic data, to perform multiomics analysis in order to understand better the profile and the characteristics of ALS patients in Egypt in comparison with other populations. And of course, we need to correlate all these omics data with the clinical data, and most importantly with the epidemiological data, in order to study the local characteristics of the disease and to look for some risk factors or potential therapeutic strategies. And that's why we are now constructing a registry for motor neuron diseases in Egypt. And this was an initiative that was launched last year in December 2023 that includes several centers from Egypt. So it's a multi-center study to collect ALS patients across Egypt, to generate the first incidence and prevalence data, and to study the epidemiological aspects of the disease. And this initiative has been recently published in Neuroepidemiology journal, and these are actually the main collaborators with the registry. Professor Albert Ludolf, he's the head of neurology department at Ulm University, and has spent his entire career searching for doing ALS research. Professor Nagia Fahmi, she's the head of the neuromuscular unit at Ain Shams University, our main collaborators. And Professor Giancarlo Groscino, he is the head of the Aging Center in University of Bari, Italy. He's a neurologist and expert in neuroepidemiology with worldwide experience in establishing registries for rare diseases, because again, it is challenging because we are talking about rare diseases. And I'm happy that we have actually constructed or established a transnational research cluster dedicated to study the ELS, rare and complex disease. This research cluster comprises several neurology centers from Egypt. We have within the GOC, our universities, we have collaboration with other departments, including computer science, and we are happy to have to be member of the GARN for the bioinformatics and AI aspects of our research. And we have also the collaboration with Germany and Italy. And I would like to acknowledge all my collaborators and my research team. It's a long journey. It has been a long journey for us. We dedicated our research to this challenging disease, and we have a long way to go. And we hope that one time we will be facilitating the treatments or making the treatments more affordable and accessible for Egyptian patients and patients in Africa in general. And with this, I thank you very much for your attention.
so much, Professor Nabila, for this nice presentation. It's very interesting, very informative. So, because we are doing it, we provide only time for one question. Attendees have a question. So, if you don't have, because we are ready, we can go to the lunch.
I think we are hungry.
We will have time for 1 hour, 3 minutes, we have to send that. And if you have any questions, thank you so much.
S'il vous plaît, il y a un peu de